Hi, everyone. So it's a pleasure today to introduce our new product called Illumina Protein Prep. So I'm just going to spend about 15 minutes talking firstly around the SOMA technology, which is the backbone proteomics technology that this product is based on. And I'll show you some data, some from inside Illumina and some from our early access customers that demonstrates the functionality and performance of the product. Okay, so just by way of introduction, Illumina has had a long history of products for genomics and transcriptomics, not just our sequencers, but also many different assays and library prep kits, as well as epigenomics. But we're really excited to add proteomics to our wheelhouse. And how we've done that is through access to SOMA technology, and I'll explain a bit more about what that is and how it works. As Anna said earlier, we think by adding additional omics, including proteomics, users can study whole biology in a much more simpler way. This particular product, Illumina Protein Prep, is an end-to-end automated workflow from sample to data. I'll show you a little bit about that. It's compatible with our NovaSeq systems, and we also have secondary and tertiary analysis for proteomics data and indeed multi-omics data through a software called Partec, which I won't talk in great deal today about, but just to make you aware of that. Okay, so let's get into the technology. So SOMAmers are a kind of aptamer, which is a oligonucleotide, which is essentially selected in a selection process to bind to a protein with high specificity and high sensitivity. And SOMAmers are a particular kind of aptamers called slow off-rate modified aptamers that actually came from a company called SomaLogic. And essentially how their technology works is rather than just using regular DNA bases, they modify some of the bases in the aptamer with different modifications. And what this does is essentially makes the oligonucleotides a bit more protein-like, and that enables them to bind to proteins with high specificity and sensitivity. And here you can see one structure of one in a complex with a protein. The aptamers themselves are utilized in an assay called SomaScan to make highly multiplexed proteomic measurements. And just briefly, some great points about these aptamers is they're highly scalable, they're smaller than antibodies, and so you can put many of them into a single reaction, and that's really one of the hallmarks of the SomaScan method. They're cost effective to produce and use. They have really good, remarkable specificity, and they're highly reproducible. And I'll show you some of the data that exemplifies that. So what about the SomaScan assay itself? It's really a set of conversions that we do in the assay. And it's a bead-based assay which runs on a automated platform so that you get the best reproducibility and can maximize the sample throughput. But in essence, the assay converts protein abundance into nucleic acid abundance, or in this case, it's the SOMAs themselves that serve as the conversion step. And how it works is you take your sample, so in this case, it's a blood plasma or blood serum, and you incubate it with some structavidin beads, as shown on this first panel on the left. And the SOMAs are bound to the beads already, and the proteins, under careful conditions, are allowed to bind to their respective SOMAs. The next step is that the proteins themselves are biotinylated, so those little blue blobs that are now added on to the proteins. That happens in the assay, and the SOMAs are actually cleaved from the surface of the beads using UV light, which happens on the TCAN deck of the automation platform. And then what happens is we add something called a polyionic competitor, which is a bit like if you're doing a blocking for a Western blot or something like that. You want to add something that makes sure that the protein is bound to the correct SOMA. So if in the case of this third protein here, the polyonic competitor elutes that off because it's not bound to the correct soma. So that's a really important part of the assay to ensure that we have specificity. And then the clever part is we recapture only the somomers that are bound to a protein because we capture them via the protein. So that's our final step here. So conventionally, the SomaScan assay has had a microarray readout, but what we have done at Illumina in collaboration with SomaLogic is convert the readout to an NGS sequencing-based readout, which has a number of advantages, including batching and exploiting the really great low cost per read of our NovaSeq series, in particular our NovaSeq X platform. So just a brief point on the specificity and orthogonal validation that SomaLogic have done on these aptamers. So firstly, in order for an aptamer to make it into the assay, it has to go through a number of rigorous tests, such as measuring the equilibrium binding constant of a purified protein, and then also checking that the assay itself, the SomaScan assay, responds to proteins by doing titrations. But that's great, but really to gain evidence that the aptamer, or the somomer, is binding the correct protein, having some kind of orthogonal or different assay to prove that is important. And so what SomaLogic have been doing is gathering a very large and comprehensive list for the entire panel that provides evidence of orthogonal assays. So an orthogonal assay in this case would be something like pulling out the protein from plasma with the particular soma and using mass spec, as you heard from Clive, to give confirmation that's the right protein. Sometimes that's not possible because of the amounts of proteins that are able to be pulled out, in which case other orthogonal methods, such as looking at the data correlating with RNA expression or with genomic variants in the case of cis-pQTLs. And so what we're trying to do in collaboration with SomaLogic is just make sure that this information about how much orthogonal validation each of the somas have had is available. And more than 2/3 of the entire panel has more than one source of orthogonal validation. So we're really trying to get that information out. So just lastly, on the introduction side, so we have a complete end-to-end solution. We have the kit. So it's worth saying that at the moment, this is a 6,000 approximately unique protein targets, but we are going to upgrade the kit in early next year, and that will be approximately 9,000 proteins. You can run around 384 samples through a week. It runs on essentially an Illumina supplied TCAN automation platform. It has some custom parts on it. Sequencing is done on either our NovaSeq X or NovaSeq 6000. And then we have secondary analysis, which converts those reads into essentially normalized protein abundance. That happens. And then we have the option to do tertiary analysis with Partec, and that's where you can really start to do some powerful expression analysis, or even bring in different omic data types to bring in with your proteomics data. Okay, so just a bit more of some data from our R&D team, actually, initially on how we've converted the SomaScan from a microarray to an NGS readout. So we have a new assay, which is essentially a barcoded probe assay. So if you remember, I said that the output of the SomaScan assay is a collection of somomers that really we want to be able to measure the abundance of those somomers, because in turn you can understand the relative abundance of the proteins in your sample. And so the first step of the conversion assay is to hybridize a large collection of barcoded probes. So we have two probes, one with a capture handle on it and another one with a barcode that's specific to that somomer and therefore in turn specific to a protein. We then capture the somomers on magnetic beads and wash away the excess barcodes. And so the reason we do this is so that we can translate the amount of somomer into the amount of barcode. And then we're into a fairly familiar sequencing kind of prep, where we amplify the sample with our P5 and P7 containing PCR primers, and they also contain our sample indexes, and we end up with a index barcode library, where rather than with conventional genomic sequencing or RNA sequencing, we're sequencing some nucleic acid material from the sample. Here we're sequencing the barcode, and that barcode can be converted into a protein amount. So it's kind of useful to think of this assay a bit like a highly multiplex plate reader, where every well in the plate will have a sample barcode in its kind of XY dimension, but in the Z dimension, so within each well, we now have protein barcodes. And in the case of our upcoming product, that would be 9,000 different proteins that you'd be able to measure within a single well. And just lastly, to follow on from what Clive was saying, it's really one of the challenges with plasma proteomics is the wide dynamic range. And SomaScan deals with that, firstly, by splitting the content into low, medium, and high abundance groups. And that enables you to dilute the high abundance proteins down when you do that. So it's a kind of physical split. We've done an additional layer of dynamic range compression to really minimize the amount of sequencing reads required for this. And you can see some example of that. So here's the abundance of the somas coming out of the SomaScan assay. We've split the content further up into different groups. And what that means is we've manipulated the amounts of reads, so we're not wasting too many reads on the high abundance, so that we can spread that total amount of reads across all of the samples. And roughly it works out at around 40 million reads per sample, but it doesn't need to be exactly that. And there's a lot of normalization that happens by our secondary analysis software to make sure that all of the samples are normalized for analysis. So when we developed this readout, of course, we wanted to check that it was concordant with the microarray, which is the standard for this Aptima SomaScan assay. And so what we did was we took 85 healthy serum and plasma samples. We ran them through both the microarray and the sequencing readout. And because we have 85 samples, we can do correlations in the population. So we can essentially do a correlation for every single protein in the panel. And these are the correlation coefficients that we see. Here's an example of a very nice correlation coefficient. We get very good agreement between array and NGS. And sometimes it's a little bit noisier, but still overall very, very good correlation between microarray. And more than half of the somas in this early access version, where we're targeting around 6,000 proteins, had correlation coefficients greater than 0.9. So one other point about proteomics, especially if you're using them for discovery, as Clive said, which this product is really well suited for, is to have a high precision. And we typically measure precision using CV, which is a way of measuring the noise. And how we do that, and this is fairly standard with lots of different proteomics assays, is you tend to run multiple replicates of the same sample within a run. So in our case, that's within a plate. We call that intra plate CV, and then between plates. And the reason that's important is it allows you to measure the CV, which is really important for if you want to find differences between samples in your population. If you haven't got a big fold change, say you've only got a two-fold expression, you want a really small variance so that you can have more confidence that you're seeing something real. And here's where we're at with our early access version. We're having median CVs of under 9%. And these charts, we call them cumulative distribution frequency, where it allows you to just see the whole population. So the best somas in our panel have a CV around 5%, but a lot of the content is under 15% CV. And here you can see some of the statistics of where we've ended up with our total CV. So we're really happy about this. And this is one of the hallmarks of precision. And just to note that we're currently developing the new upgraded 9K product, and our CVs are coming down even further from this. So we're excited about where we're going to end up with the performance. So stay tuned on that. And just a last piece of internal data, one of the things that's important is to understand detectability. So how many of the proteins are actually measuring the signal. This is quite difficult to do, and there's many different ways. One way to do it is to compare the amount of signal you have relative to the buffer blank. So as part of each run, there are some no protein controls, essentially some buffers run through the assay, and that gives a little bit of signal. So you can see the difference between when you have proteins and this blank. And what we do is we run a number of samples through, so we get a distribution of a population of samples, and then you run the blank, and that also has its own distribution. And then for every single protein in the panel, we can run this distribution analysis, and we can create a line in the sand for the LOD based on some maths. We can say, okay, is this protein above or below that signal? Here you can see one example for LEC2 protein where we've got the population signaling really high above the LOD, and here's one where we're a little bit closer. And applying a threshold, we were really satisfied that we got a huge majority of the somas above that LOD ratio, and that corresponds to the 6,000 human proteins. And then into some biology. So we had a healthy population internally, and we were able to see that these 3 known menopausal markers increase in abundance with age in the female samples. And this was well known, but the important point here is that exactly the same pattern of data was seen between the microarray and the sequencing. So that again gave us confidence that the sequencing assay was representative of the proteomics of the samples. And then lastly, this was an early access customer who had run some cancer samples through the product, and the 2 protein examples here, they see increased relative counts for a number of the cancer samples that have gone through the assay relative to the healthy control. So again, more evidence that we can detect differential expression in real world samples with this product. And just briefly, as I mentioned before, the secondary analysis converts all the recounts into protein abundance. And then what happens is you end up with a list of normalized counts, and then you can take that into our tertiary analysis for looking at visualization and differential protein expression with this Partech software. And this is my last slide. So I think I've mentioned all these things. It's a streamlined workflow, fully automated. We've got really good CV and LOD numbers, which is a hallmark of the SomaScan assay. There's a big discovery power here in our early access version. We have 6,000 proteins. That is going to be upgraded to more than 9,000 human proteins. We're still working that out at the moment internally here at Illumina, and it will all be able to run on our lowest price point per read, which is the NovaSeq X25B flow cell. And with that, I think I'm finished.